So just for those who do not know AnalytiCon Discovery, the company is located in Germany. It's headquartered in Germany, in Potsdam. That's 30 miles south of Berlin. And I'm actually not in Germany. I'm in Rockville in Maryland, where I'm sitting right now. The company has around 65 employees. So the company was founded in 2000, which is weird because I joined the company in 1998, but it's a spin out from a larger organization. That's the reason why. The company is dedicated its work on natural product chemistry, so it means 3D chemistry. And we have, as you see here on the left side, you see we have different technologies in-house. That is, on the one hand, the pure natural product part, using microbial strains and plants to produce interesting, pharmacologically interesting natural products, so secondary metabolites, if you will. Have then all the technologies to do the fermentation, isolation, structure elucidation, but also the other half of the company is involved in discovery chemistry, medicinal chemistry, but also then synthesizing novel 3D libraries, which we all sell then through the ChemSpace portal. And I will, at the end of the talk, I would summarize that in one slide. So the company, the AnalytiCon Discovery, is called the Natural Product Company. Why? Because we believe in the importance of natural products for drug discovery. And yeah, okay. So, and there's always a nice review from Newman and Greg every 5 years or so, where they talk about the importance of natural products in drug discovery and for finding and developing new drugs. On the upper graph, you see that among these probably 1,700 newly approved drugs in the period of 1998 to 2019, there are almost 1,400 new chemical entities. So the rest, the remaining 300 are biologics. I'm not talking about them here. But among these 1,400 new chemical entities, 67% can be traced back to natural product. What does it mean? It could be the original natural product, which is a drug, or it could be a derivative. It could be inspired or a mimetic of a natural product. It could be a fully synthetic compound, but without the initial natural product inspiration, nobody would have found this drug. There is the remaining 33%, it's in orange here on the left side, fully synthetic approaches. But again, 67% can be traced back to natural products in this long period. When we look at antiviral drugs, the number is even higher. It's 76%, and with 24% are fully synthetic compounds. To give you some examples where natural products play a role, these are all 2017 FDA-approved drugs which have a natural product background. If we start with 12 o'clock, you see midostaurine, that's a staurosporine derivative, obviously importance in leukemia. Then on the, let's say 2 o'clock, we have valbenazine, that's a prodrug in a neurological disorder like Tourette's syndrome. It's a product because once the valine is released, then benazine is the API in this case. The next drug on the market is Simproic, that is for treatment of opioid-induced constipation, and it's obviously alkaloid-based. The next one on 6 o'clock, that's a prostaglandin derivative for hypertension. The next one is Emflaza for Marathon Pharmaceuticals. That's a glucocorticoid derivative. And the last one is co-developed by Merck and Pfizer. That is Steglatro. That is a compound which is based on carbohydrate chemistry. It is a SGLT2 inhibitor for the treatment of type 2 diabetes. And actually the first real SGLT2 inhibitor found was fluorescein, which is a natural product. That was not the basis for this discovery, but it shows you even new targets could be addressed by natural products. So there's some nice examples about the three-dimensionality and the importance in the area. There are two publications one is from 2009, the other one from 2013, with the title Escape from Flatland and the importance of three-dimensionality for the success rate of clinical candidates to reach the market. And what, in essence, what the outcome is that the higher the FFP3, so the fraction of sp3 carbons among all carbons, as well as the chirality, is that the higher this value, the higher the chance that the clinical candidates will be an approved drug. One explanation is that the higher the three-dimensionality, the more selective a compound is, because it's less promiscuous, and therefore it has less chance of having side effects or off-target effects. So that just underlines the importance of three-dimensionality in the drug discovery space. These are the libraries we are currently offering through ChemSpace. The first library is pure natural products, secondary metabolites from plants and microbes. There are close to 6,700 compounds in stock, all produced at the headquarters in Germany. 43% of the compounds are novel, so we have discovered the compounds for the first time. And I put you just some examples to show you a little bit the broad diversity of the library. The second one is a semi-synthetic library, so lead-like, drug-like compounds. In total, 32,000 compounds in stock based on 125 scaffolds. These are natural product-based libraries, but a fully synthetic approach. There's one example here on the right side with a publication from 2020. Where a group in Spain, UK worked on screening with part of our library, and they found this mannose-derived compound, which is a highly selective, very potent GSK 3-beta inhibitor, which plays a role in Alzheimer's disease. And the last library we have are semi-synthetic macrocycles with different concepts, and I will deep dive into this later. Right now we have 152 scaffolds and close to 4,100 compounds in stock. So you see already here 3 examples of macrocycles. So why are macrocycles important, and what is the current status? Here's a publication from 2017. That group analyzed all the macrocycles in clinical development and also as a approved drug. On the left side, you see the statistics for market drugs divided into oral and IV drugs. What is shown here, the outcome is that 68% of the compounds are relevant for, have been used or are in use for oncology and infection, but only 13% of all the compounds are orally available. Among those are still 60% of the macrolides are orally available. Obviously, most of them more in the infection area. But also what you can see is only 4% are de novo designed compounds, either through high super screening or by mostly a structure-based drug design. And what the authors also mentioned, that 95% are derived on natural products. So approved drugs is somehow a snapshot what happened 10, 20 years ago. If you look at the clinical candidates, you see which direction we are going. So still the focus is still oncology and infection on the right side with 66%. And but here now the orally available drugs is jumping to 41%. And one of the reasons is certainly that the novo design compounds designed to be orally available are a rising star in this whole concept. There's another publication talked about the physical, chemical, and other properties of macrocyclic drugs, and they looked at clinical candidates as well as registered, so FDA-approved drugs. On the left side, you see the C-log-B. Middle pane is the total polar surface area. On the right side is H-bond donor. And what the authors concluded that there are some guidelines, not really rules like the Lipinski rule of 5, but at least if you for drug discovery and development stay in this certain property space, then the likelihood that the macrocyclic compound will be already available is very high. So molecular weight should be, for instance, as a guideline, be below 1,000 Dalton. C-log-P should be below 10. The polar surface area below 250, and the number of H-bond donors should be below 5. So, and you know, typically the increased molecular weight is also linked to an increased lipophilicity, which can translate into better orally or bioavailability of the compounds. On this slide, I summarized a little bit the different concepts, how to make macrocycles and find macrocyclic lead compounds. On the left side, upper left side, you see biologically synthesized compounds. There's obviously historically well-known natural products from different sources, from microbes, plants, insects, reptiles, or other sources, also marine sources. There's the possibility of phage display, messenger RNA display, and peptide splicing. On the right side, upper right side, you see chemically synthesized libraries. There's mentioned the 1-beat-1 compound, the 1-beat-2 compounds. Latest development, I think now for 10, 11 years, is now the DNA-encoded libraries, and I have a few examples about them, as well as diversity-oriented synthesis. And the final concept is structure-based discovery based on, for instance, crystal structure data. Below there, you find some examples of approved drugs, macrocyclic drugs. On the left side, you see rapamycin, a natural product which plays a role in immunosuppressant agent. On the top, you see actually a first macrocyclic compound from DEL library, so DNA-encoded library from AstraZeneca. It's an MCL1 inhibitor for use in oncology. On the top right, so there's pacritinib. That's a fully synthetic structure-based drug discovery compound. What they did is through cyclization of a linear a compound found through high-throughput screening, they found this drug. On the very right side, there is ivermectin. It's a special compound because the authors, the inventors of this compound, actually they received the Nobel Prize for Medicine in 2015. This compound has also some, so the application is more as an insecticidal compound, but it also has some antiviral properties, and that actually was used. There are several clinical studies out there to see if this can either prevent or treat COVID-19 infections. But it turned out, I think in March, it came out that the FDA and the EMA is not recommending this compound for treatment of the coronavirus. On the lower right side, there's Simproic for treatment of HCV. Then another more complex natural product from actually initially found from a marine sponge, that is eribulin from Eisai, an oncology drug for treatment of breast cancer. the initial setup was to synthesize this drug is 62 synthetic steps. So I just want to shortly have one slide about the macrocycles we have found from nature. So from microbial and plant source. So you see here the FSP3 versus the molecular weight of different types of natural product. We have natural products we have isolated, all macrocycles, and they are a broad range of different diversities. On the lower left side, very simple macrocycles per definition. The ring size is more than 12, just one stereocenter. It's not awfully complex. If you look at the upper left side, first of all, it looked like a complex compound because of this high FSP3, but it contains just some carbohydrate chemistries, and this boosts somehow the FSP3. And I think every natural product and medicinal chemist could think about some modification of this compound. And then we have the other extreme mentioned on the right side, a very complex polyketide. We have a very diverse set of natural product macrocycles. 51% of all the macrocycles we have isolated and structurally identified are novel, but even within the known set, there's an estimate from a publication in 2007 that 92% of those known macrocyclic compounds, they only have been found through antimicrobial activity. That is the way in the past natural products were identified by antimicrobial activity, but nobody really knows what other different targets those natural product libes hit. And there's an excellent source just to do deorphanizing of new targets, or if other technologies failed. But I just want to mention one publication, a recent publication from this year from AstraZeneca. It's a collaboration between AstraZeneca and the University of Uppsala. What they did, they looked at natural products, libi, filtered out the macrocycles, more than close to 3,700 macrocycles, did some, removed the side change, did some clustering, but then at the end of the day, cherry-picked 41 scores, which seems to be synthetically tractable. And from those, they screened for PPI application and then synthesized an analog through de novo synthesis. And they found a nice compound you see here on the lower end for PPI inhibitor activity. But the restriction is somehow natural. The synthesis must be available. So there are other concepts how to do this synthetically from scratch. Here summarized a little bit the findings from a 2017 publication where different types of approaches have been compared. So the first approach is the 1-beat-1 compound and 1-beat-2 compounds. They are, a large number of compound synthesis compound approach possible in the millions, but it's restricted a little bit to amide bonds. They are easy to use for high throughput screening, but the deconvolution, so after finding a hit, is rather complex. In the case of the DELs, or DNA encoded libes, the same number of compounds, and I have a few examples for this. There's a diverse reaction pool possible, amide, Wittig reaction, cycler addition, and other reactions. It's straightforward, can also be used then for structure-based drug discovery. The only limitation is a little bit the chemistry, because it has to be a DNA compatible chemistry, and also the proteins must be soluble. But in principle, the tools and the chemistry possibilities with for the DNA compatible approach is on the rise. And the last approach is DOS, so diversity-oriented synthesis, more natural product inspired rather than natural product based. A little bit smaller number of compounds, and in some cases it's natural product like, so the hit rates are not the highest compared to other libraries. Here I have a few examples. This is for the DOS, the diversity-oriented libraries. This is from Lisa from when she was with Broad. They synthesized 14,000 macrolactams to different reactions from nucleophilic aromatic substitution, RCM, as well as 2+3 Huisken synthesis. And then they took 8,000 of the macrocycles and did a phenotypic malaria screen and found this a highly potent inhibitor. The other example is from Tranzyme Pharmaceutical. They created a library of 10,000 peptidomimetics here on the left side. And when they screened them for human motilin receptor activity, they found this one compound in the middle. And also they developed a drug actually out of this library, which is relevant for the GI application. Another examples here are listed are for DNA encoded and a mixture with phage display libraries. The first one is from Professor Leo from Cambridge, synthesized, one of the first ones, synthesized DNA encoded libraries, 14,000 compounds, screened them against 36 different protein targets and found potent nanomolar hits in against SARC as well as the insulin degrading enzyme as highly potent inhibitors. Another example, recent example on the lower left side, is a publication from a South Korean group, and they synthesized their libraries, more classical peptide chemistry-based macrocycles, and this is 10 million compounds. And the last example I want to bring here is a peptidomimetic macrocyclic library that's on phage display, and then they attached another DNA tag to that library. The chemistry they have done is, if you look closely, they transformed the disulfide and reacted with 1,3 ketone, and this was then incorporated the pharmacophore model by reaction with hydrazine, and yeah, then they used it for screening for different application. It's also a recent publication from 2021. There are some reviews about the DEL space. One is from 2017. It says it would be nice with a recommendation from the author to incorporate more natural product-like building blocks. The second, somehow, at least in 2016, summary was that that is because of the limitation in the reaction, but also the accessibility of simple peptides, the scaffold diversity is not the best. So we thought about how we can address this, and you see that later in my presentation. Here are some novel approaches. The first one is a structure-based drug design that is from Alessio Ciulli from Scotland. And they worked on BET degrader of the MZ1 and did a cyclization, macrocyclization, and find highly potent degrader and also confirmed that in a co-crystal of the corresponding system. The second example is from a German group. They worked on SAR-fit ligands, which are the linear the linear examples for FK, the macrocyclic compound FK506, and they did a cyclization and found high affinity binder for FKBP51 protein. The other example is a nice example where authors introduced more diversity into the scaffolds. This is based on a DEL approach, so DNA-coded libraries. Here I just marked the building blocks 1 and 2, because the third one is transforming the diversity in the side chain from the A side you see on the upper right side. In total, they did 7-step synthesis, so medium-sized complexity. With this, they created a library of 1.4 million compounds with more than 2,000 different scaffolds. And the last publication, and that is my second part, is about macroevolution, published in 2017. This is somehow our own work from AnalytiCon. So how does it work? We took a look at our natural product chemistry pool. Coming back to that later. Then we somehow synthesized natural product building blocks, 3 different building blocks. Then we used the FMOX strategy to and synthesized the linear precursor by solid phase peptide synthesis. And once it's cleaved from the resin, then do the macrocyclization. So we started with one array, which is an 8x8x8 system. We did the solid phase peptide synthesis. So these are then 512 linear precursors. And once we cleave it at a scale of around 10 milligram, we found 100 macrocycles, and this is confirmed by LCMS. So not and this microtiter plate of a cleavage from the resin. And the next step we took, again, we went back to the 8x8x8. We pre-selected 60 linear precursors, did a large-scale synthesis of those precursors in a scale of 1 to 2 gram. Did the macrocyclization, which was 80% successfully, and then based on these 50 scaffolds, then we did an array synthesis of 500 compounds. But we didn't only synthesize one array. In total, we synthesized 3 different arrays. The first one is through the cyclization method is lactamization, so 3 amide bonds. There is a success rate of 23%. Success rate is defined by the cyclization success measured in LCMS during the exploration phase. The second arrow is also 8x8x8. That is also 3 amides. That's similar success rate. And then we did another library and incorporated then oral ether, and this is then the cyclization method is nucleophilic aromatic substitution with a rate of 17%. So here this is for the array 2, the success rate for the different calculated cycles. So you see on the y-axis is the number of theoretically cycles formed, and on the x-axis you see the macrocyclic ring size. So obviously with the building blocks we used, we were aiming at ring sizes of from 9 to 21, and what you can see here is the percentage, so the success rate of the cyclization. For instance, to give you an example, the macrocyclic ring system of 20 had a success rate of 75%. It's one interesting finding, and we don't have an explanation yet, is that the 10-membered ring system did not result in any cycles. But the other finding is that the larger the ring size, the higher the success rate, and I think that makes sense from an entropy standpoint. Here you see some examples. Hopefully you can see that it's not only just a simple peptide chemistry, so we're trying to incorporate motifs and building blocks from also polyketide character, so it's a mixed mode. All these compounds are single pure compounds available for high throughput screening. And we analyzed here a little bit the principal moments of inertia, which was published in 2014 for our 4 arrays. And this triangle represents on the left the rod-like, on the lower the disc-like, and on the right the sphere-like shape of a compound. And the more sphere-like a compound is, the higher the chance that the compound can penetrate biological membranes and therefore be biologically active or biologically available. We compared on the left side the different arrays, array 1, 2, 3, with our natural product macrocycles, and you see a very similar picture. So the macroevolution library we have synthesized is diverse and also closer to the sphere-like shape than other more flat libraries, obviously. I mentioned at the very beginning that I will tell you a little bit of the building blocks. So we used 2 sources of the building blocks. One was somehow extracted virtually from natural product building or pharmacophores, and we try to incorporate them into the synthesis of building blocks inspired by natural products to have a higher chance of being biologically active. And the second source is the semisynthetic libraries we have created, as I mentioned in the very beginning. And these are intermediates, very unique, very proprietary chemistry. And then we use those building blocks in the pool to create those macrocycles. The good thing with the macroevolution libraries is the follow-up. So I just picked a random example in the middle. So you go to 12 o'clock. You can just simply do the inversion of the stereochemistry by using a corresponding enantiomer. You can also flip, or you can also do ring contraction and ring expansion by using different building blocks and different protection chemistry. It's easy to, in the first place, to remove the amide bond and replace it maybe through an amine. Let's see what effect that is, and then you can also in the side chain work on bioisosteres side chain and replacing them because of the chemistry established. That is very simple as a follow-up and for the hit exploration phase. So overall, what we did is we tried to use guidelines derived from other macrocycles in clinical candidates and drugs, but more guidelines, not rules. We tried to incorporate into this macrocycle biological relevance by synthesizing pharmacophore relevant parts from macrocycles and incorporate that into this system. We try to be more scaffold diverse and side chain diverse than maybe other typical cyclic peptide libraries. And finally, what is important as a quick follow-up is to have a tractable chemistry in place. We also have another library. Let me see if I'm running out of time. No, that's still okay. And these are 12 different macrocycles we have synthesized, like a total synthesis. Give you 3 examples. This one here is chemotype 116. We synthesized the corresponding building blocks. One, we start from meroquinine, so quinine-based chemistry. Did the reductive amination followed by RCM. So that's the cyclization step here. You see some from compound 5 to compound 6. Then we introduced the first center of diversity through palladium catalyzed reaction. And then after saponification and protection of the amine, we formed compound 8. And then we did a second diversification by introducing R2 as an amide. Another example, which is a very unique chemistry, starts with the cyclopentene diol on the upper left side. Then when we did some reaction chemistry to form the cis diol protected group, and then used epichlorohydrin to do an in situ reaction, or this compound you see here, 3A, is formed in situ, and then it is directly in the same pot synthesized, and the macrocyclization is done through compound 3B. This is actually at that time when we synthesized it, the first example of a macrocyclic sulfone amide, so where the function is incorporated in the ring system. And then we created the corresponding Libe from this building block here. It's easy. That's very simple. I wouldn't say simple, but a very robust chemistry we have established here, and it's easy also to synthesize them in final compounds in gram quantities. The last example, and that's also really the end of then my talk, is about proline-based chemistry. So we have, this is here depicted several semi-synthetic, more lead-like, drug-like compounds, but we have created 3 different macrocycles, 16-membered rings on the lower left side. Here's one example. Did the typical amide coupling of compound 1 and 2, and this is also an intermediate form for other intermediate used for other macrocycles. Then after protection and saponification, did the amide, second amide coupling, and finally the macrolactonization is done to form this orthogonal protected building block 7 on the lower right side. And then obviously, because of the different protection chemistry, then we can introduce the diversity center 1 and 2 for final compounds. So second last slide. This just shows you the semi-synthetic library we call NAT-X, so more lead-like, drug-like compounds. And in the light orange are all the macrocycles we have synthesized. Upper limit is 900. It's in the right chemical space. The macrocycles have a very same lipophilicity than the semi-synthetic compounds. Yeah, and here's just the last slide where we categorized all the different libes, the synthetic libes we have off the shelf available, and which we sell through ChemSpace, from cherry picking to the whole sets, whatever you like. So with that, I'm done with the presentation and happy to answer any questions.
Yeah, thank you, Dietmar. Yeah, please type in your questions in the questions box. I think before, we have questions from our attendees. I have a few. You mentioned that you synthesize the compounds, and like some of them they're in gram scale. But what is the typical amount you have for microcycles and other compounds in your library, the natural compounds?
Yeah. So yeah, if we synthesize a compound, typically we aim for the semi-synthetic compound. We aim at 30-35 milligrams. There are some exceptions, but most of them are in this range. For the macrocycles, it's the same for these macro X compounds. For these macroevolution compounds, the amount is lower, but still in reasonable amounts to guarantee also the resupply if somebody purchased 1 milligram. Mm-hmm. But again, on average, I would say 30 milligrams. That was we are aiming for.
Thank you. And if the compound is depleted, you can always resynthesize it, right?
Yes, exactly. Typically, how it works that we don't have to start from scratch. In many cases, we have the intermediates available, and for 2 reasons. One, we want to have a quick resupply by resynthesizing a compound out of stock by an intermediate, and the other possibility is we even sell the intermediate, so companies can do their own chemistry, more proprietary chemistry by derivatizing or introducing the second center of diversity.
Thank you. And how often do you add new products to your catalog?
So we have a biannual release. we add to the catalog every year around 500 to 1,000 pure natural products, and we add around, let's say, between 2 and 4, sometimes 6 new libes in the semisynthetic space.
Yeah. Thank you. If you were talking about nature products, I believe that the purity may vary and may be different from 90%, correct?
Yeah, so for the natural products, the minimum we offer is 70%, but the majority of the natural products have a purity of, I don't know, top of my head, the average purity, but it's above 90%.
Thank you. And the purity is assessed by LCMS, correct?
For the natural product, it's by LCMS ELSD. That's the more relevant detector rather than UV, but it's supported by NMR.
Thank you. So you mentioned that the major facilities are located in Germany. And do you have a library stored in the US, or everything is shipped from Europe? And if this is true, what is the typical delivery time within the US?
Yeah, so we don't have anything available here in the US. It's just me here in office. So we will ship it from Germany. The shipping time, I mean, there are 2 factors. Number one is how many compounds selected, because we allow cherry picking, obviously, and we do customization anytime. We don't have anything pre-plated with one exception, but the rest is just customized to the client's needs, whether it's the format, the outline of a microtiter plate, or the amount, whether milligram, micromole, DMSO, stock solution, whatever. And the second factor is typically the US customs. Sometimes it just slips through the customs, and sometimes it takes a little bit longer. I think, let's say, for a decent library, let's say of 500 compounds, let's say 100 to 500 compounds, the delivery time is maybe 3 to 4 weeks.
Yeah. And what is your policy regarding the purity of the compounds? Do you typically reassess it before shipping?
Yeah, so we frequently analyzing the compounds before we ship them out. As a rule of thumb, if a compound was stable for 5 years and the last QC was a year ago, we think we don't have to QC it again. But if it's longer ago, the last QC was 3 years ago, we QC the compounds first before we ship them out. And we have to make sure that it's good quality compounds are leaving our headquarters. For resupply, so if somebody will ask for a resupply because of a hit, we always QC them.
Okay, thank you. One more question. Yeah, you mentioned that you synthesize the products, but I believe you work with custom synthesis related projects as well.
Yeah, absolutely. It either could be as a follow-up from our own chemistry, or could be, let's say, more complex chemistry, whether it's total synthesis or making small libraries. Anything is possible here. Yes.
Thank you. Yeah, there's one question related to ordering the compounds. So when ordering a set of compounds, is there a set discount?
Yes. So the pricing is structured as the following. So we don't have individual compound prices. It's just more or less one price fits all. Means there's a certain set of the price per 1 milligram for the macrocycles, one for the semisynthetic NADX library, and there are 2 prices for the natural products. But we don't make a difference between compound A and compound B within that group. The larger the quantity and the larger the number of compounds, the larger, the higher the discount per milligram.
Thank you. Yeah. I think one more question, for microcycles, yeah, that they've been receiving more and more interest because of their properties, and obviously you've seen the successful stories, when the customers come and basically request the heat resupply. have you seen in the trend, for example, you know, if we're talking about, you know, 10 years from now, 5 years from now, and actually now, is there a change and the interest, you know, for microcycles is growing?
Certainly, the interest in microcycles is growing, also because of the tools available to the medicinal chemist for the follow-up from the libraries what we have designed. And you know, if you compare design now and let's say 10 or even 15 years ago, it totally changed. So it's not part of the talk today, but we, for instance, incorporate a lot of carbohydrate chemistry, which is very unique chemistry. It's not easy to do, but we were successfully creating novel libraries around this. So we're trying to move in different chemical space beyond what we have already synthesized, and try to also to fill some gaps in corporate library collections.
Thank you. Yeah, I think with that, I don't have any other questions. Oh yeah, thank you very much, Dietmar, for really nice talk. So if you have any questions to to Dietmar, please contact him directly, or you can send them to me, and yeah, I'll forward them to Dietmar. Also, if you're interested in more webinars from ChemSpace, please follow us on LinkedIn, Twitter, Instagram. Thank you very much for attending our webinar, and with that I would like to close the event. Have a good one.
Yeah. Thank you, Yuri. Have a good afternoon or evening. Bye.